Let me tell you, I am a product of DAVV. I did my master's from DAVV long time back, and I see myself right back on the back benches, sitting with friends, listening to the talks, half going inside, half in the talks. So I understand where you are. So let me tell you, life is a journey, and so is research. The work that I'm going to show you is part of my journey, and it started as a side project, but now it is becoming my goal. So I hope to excite you with showing these results and sharing a very little part of my journey. So this title you think is like, it's very complicated. But when I will explain it to you, probably you will understand that it is not as complicated. Research comes in a step. You get a result and then you ask next question and then make a journey of it. So I'm coming from University of Toledo. I'm sure Dr. Tiwari, Dr. Fred, Scott have shown you. But one thing I would like you to pay attention is on this beautiful picture here. It's on the river called Moby River. The city is beautiful, has 4 seasons. You should all aim to do your PhD there. It's a wonderful place to be. My lab has 2 primary projects. One on 1433 zeta, which you see on the left, and on the right you see a SARS-CoV-2 monitoring. So first let me tell you about the project on the right. I am a principal investigator on a project in which we monitor COVID-19 spread among the students who are in the dorm, and we report that number to our county, our city health department, and based on the trend that we are observing, city officials make a guide, use that as a guide to make policies to see whether vaccination is required or other things are there. But the story that I want to share, which is my passion, is about this protein called 1433 zeta. It's a small protein, around 28 kD protein, 9 helical chains. In human, there is one gene, 23 transcripts, of which we do not know the function. There are 10 pseudogenes in addition to it, which are very, very recent finding, and we do not know the function. But the one we know, this protein has intracellular localization as well as extracellular. So when I say intracellular, it has certain set of functions like scaffolding, adapter, chaperone. It also acts as a cellular signaling protein. But once it is outside the cell, it changes its properties. It becomes like a paracrine factor. It goes out, provides the signals to other cells. And what we have shown is that it is also an antigen. So there have been a lot of studies on this protein. It is very well known in cancer. But my interest is not in any of those functions. I am interested to understand immune functions. And to understand that, I have addressed 4 primary parts. Its antigenic properties, its signaling function, what does it do at genomic level, and what does it do at metabolomic level. So together, we are trying to understand it. We have published a lot, but these are 3 findings that I feel very proud of. The very first finding that we published in Arthritis and Rheumatology was that in human patients who are having aortic aneurysms, this protein is secreted out and act as an antigen. The second paper, which we published in PNAS 2020, is showing its signaling role in the interleukin-17 signaling. And I'm sure you all know a little bit about the immunology. So in immunology, we have a lot of cytokines. Some are pro-inflammatory, some are anti-inflammatory. And in autoimmune diseases, we always think of pro-inflammatory cytokines. And interleukin-17 is one of the cytokines which is very much associated with autoimmune diseases. And the third paper that I would like to mention here is the one we published in 2021, again in PNAS. where we showed that this protein can inhibit the arthritis. And so this will be my primary story that I will tell here. So what did we show here? Does it work? Okay. Hello? No? Because I don't like to stand and present. Hello? Can you hear at the back? Yes? Okay. So let me come here. So in this paper, there were two things that we did. We used this protein and knocked it out from the animals. So there were the animals that did not have this protein. And we then subjected them to two different models. No? Can you hear now? Yes. So we tested these animals in two different models. Thank you. Hello? Okay. Forget it. I'll do it. So we tested these animals in two different models. And then what we did, which was very, very novel, we created a vaccine. And we showed that this vaccine can prevent the rheumatoid arthritis. So when we published this story in 2021, we also did not realize the impact of it. We published it, and next we knew that we were all over the news, all over the world. BBC radio, Canadian radio, all countries, hundreds of news.
Hello.
Okay. Hundreds of news portals. It was all over the world. And we were scrambling, like, what did we do? What is in there? It was just a research paper, come on. And then we realized that what we have done, what we have shown to the world. And the beauty of this project was that rheumatoid arthritis, which is a very common disease. Have you heard of rheumatoid arthritis? Raise your hand if you have. Yes. In India, it's very common. You see disjoints, inflammation of joints, and this is not a disease which is new, unlike COVID-19, which came 2 years back and we are now over it practically. But rheumatoid arthritis has been for centuries. You see old scriptures, you see that mention of it. You see mummies, you see the presence of rheumatoid arthritis. So a disease such old disease, why there is no current treatment for this? What is the issue there? We know this disease can kill people. 1/3 of rheumatoid arthritis patients die of the cardiovascular issues. Half of them have to go through the joint replacement surgery. It's severe. It affects your quality. You cannot, it has such a strong burden on the society because you are not able to hold stuff. You are not able to do your chores. You cannot do your work. So it's a big, big implication. The current treatment, what they do is just suppress the immune system. So you take the steroids, suppress the immune system, it relieves your symptom, and you're good to go for some time. But the problem lies is in the relapse. Almost 12 months, an average, and the relapse happens. On the top, the cost of it. So it's such an important disease, expensive disease. So why there is no therapy for it? So what we did, before I jump to the vaccine result, I want to show you. First of all, if you remove this protein from the animals, so what we did, we create a knockout animals using CRISPR-Cas and subjected these animals to the rheumatoid arthritis model. We used two different models, and I would like you to pay attention to this red line, which is a knockout. And you can see the y-axis where the rheumatoid arthritis scores are, it's very, very high. So once the protein is not there, the animals are severely susceptible to the disease. We look at the pictures, you can see the joints are very inflamed. You see histology, again the same. You see the bone. Bone seems to be eroding in the case of knockout. So that was the first indication that this protein is doing some function in the rheumatoid arthritis, and probably it is having a preventive function. So once we knew this, the next question was, where do we go from it? Now, as I said, my interest is understanding its immune function. And I knew from my previous studies that this protein is an antigen. So in human patients, not even patients, all of us here, healthy, non-healthy, we are all having this protein in our plasma. We are having antibodies against this plasma. What is happening is, in the different diseases, its level goes up or down, and that changes or that brings the disease. So once we knew this protein is an antigen, and we knew that if you take the human immune cells and put these cells with this protein, the cells will start differentiating into the Th17 or Th1 cells. So we were sure that this is an antigen. And see, you always know, you do the experiments, you find results, you make your hypothesis. So based on these knowledge, we hypothesized that if we put this protein as an antigen into the animals, the animal should be protected. So that is exactly what we did. We made purified protein, we mixed with adjuvant, and we injected in the animals. We use a very traditional 2-booster shot strategy. We injected, we started the disease and gave them the 2 shots of vaccine. And what we found here, again I would like you to pay attention to the red line, which is the immunized animals. We saw the disease was suppressed. Again, the histology, the visual, everything was saying that disease is gone. This was very, very interesting. If you look at this bar, it tells you that antibody production was very, very high in these animals. Again, suggesting that this protein is acting as an antigen. So it is good for you, and you can use this, its antigenic property, to suppress the disease. Very, very good. But the question was, you are testing in a transgenic animals, which is not the wild type like we people, right? So our results can be really skewed. So we had to show this in the wild type animals. So we again tested in the wild type animals, and we saw the animals which received this vaccination, they resolved the disease very quickly. Again, supporting that Zeta is helping us. Side effects. Any vaccine you take, there will be some localized effects, right? There will be a little bit of pain, there might be a little bit of fever. But other than that, we did not notice any significant effect or on the animals. There was no visual thing that we could see the animals is having any stress. However, we did see that the Th17 cells were up. We also saw the antibody production was up. So antigen is working, but it is not giving any visible side effects. Then the important question was, as we think, if it is an antigen, it is producing antibody, and those antibodies are protecting us, like it happens in the COVID vaccine, right? We all took the shot, the antibodies were produced, and those antibodies counteracted the virus, and we were all protected. So that was the same question that we asked. What happens here? Is it the antibody that is working? So we did a passive immunization. We isolated the antibodies and gave the shot to the animals. Again, this was, although a very surprising finding, we found these antibodies are doing no effect. There is no effect of these antibodies. So it is not the antibodies that are protecting. So that brings us that it has to be antigenic properties. Now, antigen-based immunization giving us immune protection is a very hot field. It's called antigen-specific tolerance. So this is where we are now moving to. But importantly, what we found that these animals, when they are receiving the immunization, they are improving their bone quality. So if you take the long bone and make cross sections and see what is the bone doing. So this is your cortical bone and this is your trabecular bone. So that is preserving. Similarly in the wild type, again the tissue mineral density is strong, so bones are becoming stronger when these animals are given vaccination. So this tells us that the impact of this vaccination can be beyond rheumatoid arthritis. We can extend it to other diseases or other bone diseases. Now, once you have this kind of finding, you start to wonder, if this is such an important protein, so why nobody else noticed it, right? Why am I the first one to notice it? It doesn't make sense. Means people have been studying this protein and rheumatoid arthritis for decades, centuries. But, they say that discovery is made when everybody is watching, but you are thinking. So we went and started scanning through the literature. And we found two very important papers. One published in JCI, where they showed the people, rheumatoid arthritis patients who resolve this disease have very high level of this protein. And this is the only protein that correlates and is statistically significant for resolution. The other paper, which was published a few years back in Nature Medicine, said that B cells secrete this protein, and this protein is secreted less in rheumatoid arthritis. Again, supporting our hypothesis. So we are very, very confident that we have really hit a gold mine there. So what is our current model? We believe that in our body, if this protein is going down, we develop RA, and we can bring this protein up, its antigenic function up, by giving this vaccine. And we are calling this vaccine as a Zeta vac because it's on the Zeta protein. This is patent protected, obviously. So now the major question that we are trying to address is that what is its mechanism of action? So we are seeing two major functions with this protein. It is anti-inflammatory and it is bone preserving. So anti-inflammatory function, we have certain clues. Obviously we do not know the mechanism, but we have certain clues which we are now working on. The one clue is the interleukin-1 beta is suppressed with this protein. How many of you know interleukin-1 beta? Does anyone know what does interleukin-1 beta do? Have you heard of pyrogen? What does that do? It changes the body temperature. Exactly. So interleukin-1 beta is a one cytokine that comes up when we are having fever, right? So this is a cytokine which is responsible for it. And this is a very, very pro-inflammatory cytokine. So its level is modulated by this protein. Another observation that we have already published is it is bifurcating the interleukin-17 signaling, which is again a very pro-inflammatory cytokine and associated with autoimmune diseases. And in this particular disease, in this particular cytokine, it suppresses the CXCL production, which is a chemoattractant for neutrophils. So the inflammation will not occur if you suppress the neutrophil immigration. So these are the two mechanisms we believe it is acting upon. And on the bone, what we have found that if you immunize the animals, there is more collagen production. So if you produce more collagen, what will happen to your bone? Is collagen important for bones? Yes, because collagen is a matrix on which the calcium phosphate will be precipitated, right? And it will be solidified. So if there is more collagen, there will be more solidification. And that is what exactly we are seeing. So all the blue color that you are seeing is increasing when there is 14-3-3 setup. And we isolated the mesenchymal stem cells, incubated with the protein, and we see the collagen production goes up. So there is more collagen, more of the strength. So our model is that 1433 setup is changing the bone homeostasis. It promotes osteoblast. It makes more bone. It suppresses osteoclast by suppressing these all inflammatory cytokine production from the synovial fibroblast. So currently we are working on the mechanism of action. Now to take this vaccine to the clinic, we have to cross several steps, and some of them are we have to do the safety and toxicity analysis. Before we inject this into any human being, it has to be approved by FDA, and there are several regulatory steps that we need to cross. We have to make the human grade protein and test its therapeutic ability. So this is my team, my students, my master students, and the work that I presented today is primarily done by these two students, Jenna and Joshua. These have been my master students who have been really very hardworking students. My collaborators from University of Toledo and outside Toledo, and my funding agencies. I really thank them, and thank you. And before you clap, students, I want you to read this quote, because this is a quote that I believe in, that our greatest glory is not in never falling, but in rising every time we fall. So remember, in life, we all are gonna fail at one step or other. That you cannot avoid. But if you are bold, you will stand up every time and move on. Thank you. I'll be happy to take any questions. So the question is, which organ is getting most affected by this vaccination? So we generally give the sub-Q. We give the sub-Q injection. We have not given directly in joints or any other place. We are seeing the systemic effect. And we have tested the effect on, it's just the histological analysis of liver, kidney, and we are not seeing any toxic effect on that and spleen. Thank you. Yes.
Is there any correlation if the level of population is high? Means like dose escalation, if you are doing it, is there any like toxicity associated with too much of this protein?
Yeah.
And just follow-up question is like, are you collaborating with any pharmaceutical company for this GMP grade production and all those things? Because that's the next critical step for your preclinical and clinical study.
Yes. Yeah, very important question. So first, to answer your first question, that what happens if the level goes very high? So let me tell you a story here. I'm sorry if I'm taking too much time, but I'll be quick. So I found this protein as an antigen in the human aortic aneurysm patients, and the level was very high. And I started this research project with a hypothesis that this is a bad protein for us. So what I was trying to do all the way was to remove this protein from the system. And I thought if I remove it, animals will be protected. When I did it, it was opposite. And I was so depressed. I was so frustrated with the result, and I said, this does not make my hypothesis. So something is wrong. I went over, and now, over the years of experience, I have realized that when it is high, it is a host response. That body is trying to fight with that, so whatever the trigger of the disease is, and that is why this protein is coming up. So, does this increase gives any toxicity? We don't think so, but it's very difficult to dissociate its level increase because of the host response, because the other trigger is still there. So it's a little bit complicated to really answer that. So I hope I answered your question. The second, for the GMP and the GLP grade, yes, we are talking to the companies. We haven't finalized the contract yet, but we are really looking, we are open to many more companies and negotiations yet. Thank you.
Thank you, ma'am. Thank you so much for your words of wisdom. It will be a great pleasure for us to listen to you again in our future events. Now, I would like to request Dr. Shikha Agrawal to present the memento to Dr. C. Karthikeyan, the session chair, and Dr. Ritu Chakravarti, the keynote speaker.